Hello, everyone. My name is Anton Petrov. I'm the RNA Resources Project Leader at the European Bioinformatics Institute, which is based near Cambridge, UK. And today I will be talking to you about databases for non-coding RNA research. I would like to start by thanking the organizers for inviting me and giving me this opportunity to talk about RNAcentral and Rfam. And if you would like to follow along, you can access the slides at this URL down here. And I'll have lots of links in the presentation. So if you want to find out in more detail about anything, please go there and you'll get access to the information. Or you can contact me after the presentation. I'll be happy to share the links. Now, I'll start by talking a little bit about my background. So I'm originally from St. Petersburg, Russia, and I got my bachelor's in biology from St. Petersburg State University. When I was choosing my specialization, I really wanted to do something different from everyone. And since people were working on, they were working in wet labs doing genetics or zoology or botany or something like that, I wanted to do bioinformatics because it was very different. When I was done with my bachelor's, I went on to the States to join the Leontis lab at Bowling Green State University. And there I was working on RNA bioinformatics. And that was why, because everyone was working on genomics or some kind of DNA or protein bioinformatical projects. So I once again wanted to be different. And so it was RNA bioinformatics for me. So I'm actually very happy that this is how it turned out, because RNA is a very exciting area of research and there is just so much to study. And I'm really happy that I came across this almost by chance. Now, one of the important takeaways from my graduate studies was the importance of the non-canonical RNA base pairs. And when we talk about the differences between RNA and DNA, we usually talk about the AT and GC base pairs in DNA. Although there are other kinds of base pairs, but they're not as common. In RNA, at the same time, there is just a whole zoo, a whole explosion of different kinds of base pairs. And there is a whole classification of 12 geometric families of base pairs developed by Leontis and Westhoff. And if you think about RNA base as a triangle that has 3 different faces, then when 2 of these triangles come together, then there are lots of different ways in which they can interact. And so this is what creates this exciting diversity in RNA structure. Not to mention that if you have 3 or 4 different bases coming together, forming triples or quadruples, it becomes even more exciting. If you'd like to take a look at the interactive examples, you can go to the RNA base pairs catalog and play around with it. But I'll move on and talk about RNA 3D motifs. So these structures are organized by non-canonical base pairs. And here we're looking at an example of a triple sheared motif from Thermus thermophilus small subunit ribosomal RNA. Now, this exact structure, although with a different sequence, but with the same base pairs, with the same pattern of base pairs, is found not only in small subunit, but also in large subunit ribosomal RNA, in riboswitches, in group 1 and 2 introns, and in RNase-P, lots of other RNAs. So there's something very special about the specific arrangement of base pairs in the specific RNA structure, that it occurs in so many RNAs independently. Now, the cool thing about the 3D motifs is that they present this exciting opportunity for hydrogen bonding. Whereas the helix is very uniform and is just the same type of chemical environment, the 3D motifs, they are really special, and the combination of these specific arrangements make them very attractive targets for binding proteins or perhaps other RNA motifs or other molecules. So this is why I was interested in 3D motifs. And in addition to that, there's also the conservation. If you look at the ribosome, you will see that 3 motifs are highly conserved. For example, 22 out of 32 hairpin loops are conserved in structure between bacteria and eukaryotes. And of course, there's billions of years of evolution between bacteria and ribosomes, but the 3D structure of these specific motifs stays the same. So it means that they're really important, and we need to study them more, which is what I did. And we created something that's called RNA 3D Motif Atlas. So our goal was to automatically extract and classify internal and hairpin loop motifs. And as new 3D structures are available, they are analyzed and added to these 3D motif families. The exciting reason for doing this is that now we can use the 3D motif groups as training sets for training special software so that we can predict these 3D motifs from sequences. And this is done by a program called JARED, and there is a web server, and you can find out more about it in this paper. So this is the gist of my work in graduate school, but something that I kept noticing again and again was just how difficult it was to access information about non-coding RNAs. For example, if you wanted to just download all RNA sequences with experimentally determined 3D structures and then filter them by RNA type or by organism, or if you wanted to do it programmatically, or if you just said, just give me the reference sequences from E. coli and human small subunit ribosomal RNAs. At the time, when you were looking for information like this, it wasn't straightforward at all. And one day I came across this paper about RNAcentral, and that was a database. The idea was that this database would bring together all this information in one place, and it would be a one-stop shop for all RNA information. It was such a great idea, I thought. There was only one problem. RNAcentral didn't exist. It was just this paper, and there was no website or no database yet. And in a crazy coincidence, just a few weeks later, I came across this job ad where the Bioinformatics Institute, European Bioinformatics Institute, was looking for someone to join the RNAcentral project as a bioinformatician. As you can tell, I was very excited about this prospect. And I was fortunate to join the team and move to Cambridge to work on creating RNAcentral. Now, I would like to talk to you about RNAcentral and give you a quick overview of the RNAcentral database of non-coding RNA sequences. RNAcentral was launched in 2014, and to date, it brings together under one roof over 20 million sequences that represent a broad range of non-coding RNA types and different organisms. There are over 40 databases that are specialized in different types of RNAs or in different organisms that are all brought together in RNAcentral. And so whether you are a computational biologist or whether you are a wet lab biologist interested in just a handful of RNAs, you can always come to RNAcentral and find the most up-to-date comprehensive information about the RNAs you're interested in. When you go to the RNAcentral homepage, it's quite easy to get started. You can either use text search, sequence search, or a genome browser. So let's look at these in more detail. Here's an example of a sequence search interface. And here you can search by pretty much anything you can think of. It could be a gene, it could be species, it could be RNA type, for example, microRNA or snRNA. It can be accession, for example, a PTB accession, or pretty much any keyword that you can think of. And the cool thing is that you can filter all these results by using the facets. For example, you can filter by RNA types, by organism, or by a database where the sequence comes from, and then you can just click and go there and find out more about this RNA. And of course, the results can be downloaded for further processing. The sequence search is another cool tool that RNAcentral provides. And before RNAcentral, something like this didn't exist. But now you can go and search a really large collection of RNA sequences in one go, in one place, and see whether your sequence is similar to any of the known non-coding RNAs. Just like with text search, you can also explore the results using facets, or you can type any kind of query to filter the results. So if you're interested in a microRNA from a special organism, you can identify the similar sequences very quickly. The search is powered by NHammer, which is a nucleotide version of the Hammer search engine, and it's more sensitive than BLAST, so it's easier to find things in RNAcentral using even short queries. And in addition to just the similar sequences, you can also view some additional information, such as the secondary structure or the RFAM classification. Finally, let's talk about the genome browser. And what's interesting about integrating so many different databases is that we can do additional analyses once we get the sequences. One of the things we do is we automatically map all the sequences back to the reference genomes where they came from. And that enables really cool discoveries or rediscoveries of sequences. For example, we worked closely with the Flybase database, which is the authoritative resource annotating genes in Drosophila melanogaster, the fly model organism database, fly model organism. And there, by comparing RNAcentral with what was already known in Flybase, we were able to identify some SNRNA genes that were not in Flybase yet, but were in RNAcentral. And we did it by comparing the genome annotations in both resources. So it could be a quite powerful way of identifying something that you would not necessarily come across unless you aggregated lots of information and performed all this mapping. Here's an example RNAcentral entry. And here we're looking at the microRNA precursor. So we have all these links to all these databases that I've been telling you about, where you can see the detailed information, including links to diseases, for example, from Malacards. And you can see the genome location and other RNAcentral sequences that are found in this region. You can see the gene ontology terms that have been either automatically or manually connected with the sequence. And this is very cool because here somebody actually manually read a paper, went to RNAcentral, and then associated this specific RNA sequence with positive regulation of nitric oxide by a synthetic process. So now we know that there is some connection, some functional association between this sequence and this biological process. And as time goes on, more and more sequences will be associated like this and annotated like this. Now, last but not least, we also have publications. And in the next few years, we're going to make this section even better because we'll be automatically annotating, we'll be automatically linking the papers that just come out in the literature with the corresponding entries in RNAcentral. And that will be very powerful. It's not available yet, but stay tuned. Depending on the RNA that you're looking at, you will find additional information on different types of pages. So for some sequences, you will also see secondary structure visualizations. You will see orthologs, paralogs, RNA family classifications using RFAM, modifications, conserved structural motifs, and even intermolecular interactions. So for example, you can see which microRNA interacts with which long non-coding RNA, or which microRNA regulates which protein, which is also something that you can see directly in RNAcentral. There is lots more to explore, and we recently published a series of protocols describing exactly how to use the text search, sequence search, and there is in-depth information about how to access the data programmatically using bulk downloads or the APIs, or perhaps even direct access to the database. Now, I would like to draw your attention to Galaxy workflows for RNA-seq analysis. This series of Galaxy workflows that we developed allows people to just plug in their RNA-seq data sets and see which non-coding RNAs annotated by RNAcentral are differentially expressed in different conditions. And that can be very powerful because all you have to do is upload the data and click run. And sooner or later, you'll get the results about the differential expression of non-coding RNAs. You can find more information in this paper down here. I also want to point you to the latest RNAcentral paper that describes the most recent developments, such as the sequence search, the transition to sequence ontology to accurately annotate non-coding RNA types so that you can say that this specific RNA is not just tRNA, but the alanine tRNA, or that this ribosome RNA sequence is specifically small subunit mitochondrial ribosome RNA. So things like that. And you will also find about some exciting secondary structure functionality. When RNAcentral was first established, it contained just the sequence information that we imported from different member databases. And so we knew that something was missing, and the users were telling us that they were really interested in the secondary structure. So we knew that we had to do some additional work to visualize secondary structure. And for that, we developed a special software called R2DT that I'm going to tell you next. Many RNA sequences form really intricate and complex secondary and 3D structures. And typically, they're visualized in certain orientations that have been accepted as standard by the community. So if you work with ribosome RNAs, it's guaranteed that you will have seen structures similar to this one. And the reason for that is not because they look pretty, but because when you put two or more structures side by side, you can compare them and quickly see differences and similarities, even between structures that are from bacteria and human. So there are billions of years of evolution between these two structures, and yet you can immediately recognize similar and different elements in these two structures. For example, here's an expansion segment that we see in human small subunit ribosome that is not present in bacteria. Let me give you an analogy. Here's a map from a recent British election. And if we focus on this top part here, we can see that it's blue in the map on the left, then it becomes orange in the middle, and pink on the right. And it doesn't really matter what the colors stand for, but it's very easy to look at them and compare the differences. Now, if we suddenly flip and turn the maps, then it's not nearly as easy. So let's see. Here's the blue region at the top. Then here it is again, and it's orange. And then we have to go down here to see that this is pink now. So hopefully it convinces you that you really need to be looking at certain things in certain orientations to make them useful. However, generating RNA secondary structure diagrams in these useful orientations is not something that's possible to do automatically, at least not until now. So here's a manually curated layout. So we want to take a sequence and make it look like that. And this is what our pipeline does. So this is the result of taking a sequence and running it through our software called R2DT. And we get the diagram like that. That's great. However, other software doesn't generate as useful of diagrams as we see here on the right. And I have to emphasize that the sequence and the secondary structure is identical in all these 5 cases, but only one of the diagrams is actually useful. Here's an overview of the pipeline for R2DT secondary structure visualization. And R2DT stands for RNA 2D templates. So at the heart of R2DT pipeline is a big library of templates that contain over 3,600 different RNA layouts. A template tells you exactly where each nucleotide should be positioned in this diagram. So it defines the XY coordinates of each nucleotide, as well as the secondary structure associated with this RNA. So what we can do is we can automatically select the closest template for a given input sequence using Ribovore or tRNAscan SE 2.0 software. Then with Infernal, we fold the sequence that is given to us into a structure that's compatible with the template from our library. And then finally, we can visualize all of this using Traveler to generate a secondary structure diagram. Where do we get the templates? So for that, we worked with lots of people around the world, and we have manually curated templates for many types of RNA, the ribosomal RNAs and tRNAs. And we also have a large number of templates for small RNAs from RFam that have not been manually curated. And we'll see examples in a minute. So to start with, I want to show you the same secondary structure of small subunit ribosomal RNA. And then I want to illustrate that you really need to have a lot of different templates to represent the differences, because here's Thermus thermophilus and here's Plasmodium falciparum. And you can immediately see that there are some regions that are different between the different species, and they're species specific. So this is why you need to have lots of different templates. And what you also want to do is, when you have a 3D structure, you can look in detail at these species specific regions, and you can reconstruct the exact secondary structure and then include it in the template. So for example, here we have a 3D based template that provides the detailed description of the positions of each nucleotide in the species specific region that is not available in some of the other templates that people have used before. So this is something that we've done specifically for R2DT. The crown jewel of the method is the ability to generate secondary structure in these beautiful, useful layouts, not only for small RNAs, but for some of the biggest structured RNAs known to date, such as the large subunit ribosomal RNA. So here we see human large subunit ribosomal RNA, and you can see these vast expansion segments. This RNA is over 5,000 nucleotides long, and yet R2DT automatically visualizes it using the R2DT pipeline. On the other side of the spectrum, for very small RNAs, such as tRNAs, we also have a large number of templates. We have 74 different isotype-specific templates for tRNAs that really help model different types of variable loops and various structural features in different tRNAs. We have also templates for other kinds of RNAs, not just RNAs and tRNAs. For example, here we're comparing human and mouse snRNA. And you can see that by drawing this RNA from 2 different species in exactly the same orientation, you can just switch between them and see how these sequences and structures are different in a few locations. We have applied the pipeline to over 14 million sequences in RNAcentral. And when you run a text search, you can see the little simplified thumbnail view of the RNA. But when you click on them, you see the detailed view, and you can explore every single nucleotide. The diagrams are interactive. You can download them in a number of formats. This makes RNAcentral the largest high-quality RNA secondary structure dataset in the world. And as we develop even more templates and improve the method, we will have even more and even better secondary structures. We have done extensive validation of the method, and we have shown that R2DT templates model the conserved core of most structured RNAs. To do that, we took about 6,000 really highly curated sequences from 9 high-quality databases. So we knew that those sequences are definitely good RNAs, and when we ran R2DT on these sequences, we were able to generate diagrams for 86% of the sequences. So in the vast majority of the cases, we automatically generate a diagram. And when we manually inspected them, we found that over 99% of the diagrams were good, and they represented the templates and the RNAs accurately. But then we did a more detailed study where we looked into whether the templates and the diagrams are actually similar. And for that, we shown that 94% of the nucleotides were visualized using exactly the same coordinates as defined in the templates. Now, this is not a requirement. This is the pipeline can, of course, adjust the position of the nucleotides if necessary, if there are sterical clashes, or it can even rotate the helices to avoid problems with big insertions, for example. But it was very encouraging to see that in 94% of the cases, it just worked without having to resort to these measures. The software is available as a web server. So if you go to this URL, you can submit your own sequence and visualize it using this method. You can also use it programmatically. You can get the code from GitHub and make improvements if necessary, or run it in your own machine. And another neat thing is that you can actually embed a diagram like this on your website by just using a few lines of code. So if you want to include it in your blog post, or if you maintain an RNA database, you can just include them without having to run anything at all. The details of this work are laid out in this preprint that's available on bioRxiv today. And we're hoping to have the full publication out in the next couple of months. The future plans for R2DT are very exciting to me personally, because as I mentioned in the beginning, I'm very interested in the connection between the secondary and the 3D structure and the non-canonical base pairs and 3D motifs. And this is exactly what we can visualize with R2DT, because here's a diagram that I was showing in the beginning. These loops, they are part of the R2DT diagrams, and we can connect them to the motifs that they correspond to using RNA3D Motif Atlas and Jared and other tools. Now, in the last part of today's talk, I would like to tell you about one more RNA database that I'm contributing to. It's called RFam, and it's the RNA families database. In RFam, each of the RNA families is represented by several things. It all starts with a multiple sequence alignment that brings together all the known instances of a certain RNA. For example, a tRNA multiple sequence alignment will include tRNAs from human, E. coli, and other notable organisms, all aligned to each other and annotated with a consensus secondary structure. So a structure that's shared by every sequence in this alignment. We use the alignment and the secondary structure to build a so-called covariance model. Covariance model is a mathematical representation of the information stored in the alignment and the secondary structure that allows us to find other instances of the same family using special software called Infernal. So RFam has been established in 2002. So it's a much older database than RFam, than RNAcentral. And it contains lots of different types of RNAs, such as microRNA, small RNAs, various SNORNAs, cis-regulatory regions, such as internal ribosome entry sites, and others. And the key use of RFam is the ability to take the known families and their covariance models from RFam and use Infernal software to annotate any sequence with these RNAs. So for example, if somebody sequences a new genome and they want to find all non-coding RNAs in this new sequence, they can easily run RFam on this new genome and identify all the RNAs. And in fact, this is what people have been doing for the last 20 years. And the human genome, the octopus genome, you name it, they've all been annotated with Infernal to identify RFam non-coding RNA families. Now, here's a tweet that one of my colleagues recently posted. So he said, when I started my PhD back in 2017, working on RNA structures and virus genomes, the RFam was and is something like a Bible. And I completely share this feeling. And when I became responsible for RFam in 2015, it seemed like a very daunting responsibility to be suddenly in charge of this venerable and important resource. So today, I'd like to share with you some of the new developments in RFam that we describe in our newly published paper that just came out in Nucleic Acids Research. And in our latest series of releases of RFam database, we focused on expanding the coverage of metagenomic, viral, and microRNA families. So let's go through them one by one. Let's start with viral RNAs. One of the first things we did after the coronavirus outbreak earlier this year was to revise consistently all the RNA families that were found in coronaviruses in RFam. And this is part of a bigger project that we had initiated last year, and it had no connection to the SARS-CoV-2 outbreak. But the timing was very special. And earlier this year, we released the first set of coronavirus RNA families that represent the entire untranslated regions, or UTRs. And so we have special families for 5' and for the 3' untranslated regions for different groups of coronaviruses, such as alpha coronavirus, beta, gamma, delta. And we also have special group for sarbecovirus UTRs that include the SARS-CoV-2 virus that's responsible for COVID-19 disease. And this allows us to model the structure of these regions for the actual SARS-CoV-2 genome. You can find all this information at rfam.org/COVID-19. And in addition to coronavirus genomes, we have also done some revising of flavivirus families. So you can also find that information on the RFam website. This is all part of a bigger project, as I said, that has no connection to the coronavirus outbreak. What we want to establish is a database that will contain expertly curated viral RNA structures that have been reviewed by expert virologists, so that RFAM becomes the authoritative source of non-coding RNAs and structured RNAs in viruses. So we do it in collaboration with Manya Martz's lab. And I'd like to thank Kevin specifically for a lot of work that he's been doing. So he's the one whose tweet I shared earlier. Now, all of this work is tied together. So we've been talking about the visualization of RNA secondary structure using R2DT. And when we create new families in RFAM, their secondary structures become templates for R2DT. So when we created models representing the SARS-CoV-2 UTRs, we're now able to take the actual variants of the sequences of the virus that are sequenced by various groups around the world in actual patients, and we can visualize the sequences in the same orientation as you can see here, and compare them and see where exactly the changes are between different patients or between different related viruses. Now, moving on, another project that we're working on is synchronizing microRNAs with MIR-base. Those of you who work with microRNAs know that MIR-base is the best resource for finding information about microRNAs. And RFAM had also had some microRNA families, but they were not synchronized with MIR-base. So what we're doing now is we're working together with MIR-base to create a comprehensive classification of microRNA families that will be shared between MIR-base and RFAM, so that every sequence in MIR-base will be annotated by one of the RFAM families. So this is still work in progress, but we have been able to create about 400 families so far, and we have about 1,600 more to go. So that's going to be a big project, so stay tuned. We also worked closely with Sascha Weinberg's group at the University of Leipzig to include in RFAM all the metagenomic RNAs and various other RNAs that have been discovered by the Breaker and Weinberg groups in the last decade. And so we have significantly expanded the number of RNAs included in RFAM. So you can see some of the examples here. And also, most recently, we included some of the ribosomal leader RNAs as well. You can find more information about how to use RFAM and how to use the website, the public MySQL database, how to interpret the results, and most importantly, how to annotate sequences yourself if you want to, in this series of protocols, in current protocols in bioinformatics, similar to what we did for RNAcentral. And I want to emphasize that all this work feeds into each other. So we're talking about virtuous cycles of updates in bioinformatical resources. As we have more RFAM families, we can add more sequences in RNAcentral as we scan additional genomes and identify more instances of these RNAs. When we have more sequences in RNAcentral, we can build better RFAM families because we see more of the examples. We can review the seed alignments, include more sequences in there, and make the covariance models better. And as we have more templates, more families in RFAM, excuse me, we have more templates for our visualization software in R2DT. So to summarize, I want to say that we talked about lots of different things, starting with non-canonical base pairs in the 3D structure, moving on to RNAcentral and all the tools that it has to provide the sequence information for non-coding RNAs. We talked about visualizing RNA secondary structure using this new tool called R2DT. And we also talked about RFam, a database of non-coding RNA families, and how it expanded in several important ways to represent viral microRNAs and metagenome RNAs. All of these resources either are or will be closely linked together, and it's important to realize that only ongoing updates of all these places and establishing tight communication and connections between them can lead to new insights and new understandings of RNA in this particular case, but broadly speaking, understanding the function of molecules in our bodies, and eventually lead to understanding of disease and new therapeutics. So this ongoing work is really exciting, and I hope that I was able to share some of that excitement with you today. So I will close by thanking all my collaborators and teammates, as well as the funding sources, the BBSRC and Welcome for funding, as well as everyone in the RNAcentral consortium who provides us with the data, my team and colleagues at EBI, as well as everyone who worked on R2DT and on the RFAM projects. And with that, I'll be very happy to take any questions, and you can contact me anytime by email if you have any questions or would like to learn more about any of this. Thank you very much.